is Meryl Friedman, and I'm the Director of Education and Special Initiatives at the Center for the Art of Performance, all the way on North Campus. Anybody been to Royce Hall this year or ever? Yay. So we're the professional arts presenter on campus. We present over 75 performances each year, most of them here on campus in Royce Hall and in other venues, and about a third of our season down at Theater at the Ace Hotel in downtown LA. And my friend and colleague Sonia Luna, who works here at the Institute, introduced me to Rita Blaik almost about a year ago now. And I had never heard of the NanoSystems Institute before. And when I found out about the remarkable things that they do here, I thought of my dear friend Meredith Monk, who I knew we were presenting this year in a wonderful project called Cellular Songs. And cells is kind of the business or the building blocks of what gets worked on and looked at and researched here in this building. So Rita and I talked a bit and thought it would make for a really wonderful conversation to have an artist that we have a long relationship with in conversation with UCLA's researchers and scientists to talk about this remarkable thing called the cell. So we're going to dive in today, and that's what we're going to do. We titled this panel, Sensing the Invisible, and I realized today that that's kind of what my job is here at UCLA. If you're wondering, like, well, who is she and why is she here? My job is to kind of make the invisible things here on campus seen, not only for our audiences, but for all of us who are part of the UCLA and LA community who tend to toil away here in our silos at UCLA and have no idea that anybody else exists. So it's always an honor and a pleasure to find out what people are working on outside of the realm that I'm working in every day. So thanks all of you guys for choosing to be a part of it. It's what makes our jobs worthwhile, is to share and to explore and to figure out things that we don't know. So I came across this little quote yesterday. There's a wonderful philosopher, French philosopher by the name of Pierre Teilhard de Chardin. I don't know if anybody knows his writings. I knew you would, Meredith. Which is why I picked it. My husband has been reading his works for years, and we have lots of books of his on our shelves, and I've only kind of looked through cursorily. And in addition to him being a philosopher, he was also a Jesuit priest and a geologist and an anthropologist, and he was involved in some pretty extraordinary discoveries. And so I picked up one of his books, and I found this quote, which I thought was kind of relevant to tonight, so I'm going to read it. He wrote, "How great is the mystery of the first cells, which were one day animated by the breath of our souls. How impossible to decipher the welding of successive influences in which we are forever incorporated. In each one of us, through matter, the whole history of the world is in part reflected." So we're going to dive into that in all of our various and deep ways, and I thought that was kind of a nice way to get us started today. So for those of you that don't have tickets to see Cellular Songs at Royce on Saturday, and you're interested, and you need a little extra perk to kind of come up to North Campus, I have some 2-for-1 vouchers that I would be happy to share with you. So see me at the end, and I will be happy to pass them on. And afterwards, as you exit, if you go to your right, there'll be some coffee and tea and sweets, and we hope that you'll stay and we can have a moment to say hey to each other in a more informal setting and have a little hot drink. So I'm going to turn it over to my colleague Rita Blaik, who will guide us through the rest of this. Hopefully we'll have some room at the end to have some questions and answers. I'll be floating with the mic, and I can't thank you all enough for coming. Thanks so much for coming.
Wow, such a beautiful and poetic introduction. I don't know quite how to follow that other than to just introduce myself and our panelists. So welcome everyone who's never been here before to the California NanoSystems Institute. My name is Rita Blaik. I am the manager of education for the CNSI. My background is as a scientist. I graduated here with my PhD in 2015 in material science and engineering. And the whole time I was doing my doctorate, I realized the two things that I love the most are not only science, but communicating science, and by an extension of that, also science and the arts and the way that those two intersect. So I'm also an artist, and I always try to, in my work, communicate the amazing things that I get to learn as a scientist through art as a means of getting to communicate what that is. So it's truly an honor for me to get to have a conversation with all of these amazing artists and scientists that are here today. And I'm just gonna go ahead and, instead of me trying to introduce them, I think it's better to let them introduce themselves. So we are going to start with the person that brought us all together today, which is Meredith Monk.
There. So glad to be here. I'm so excited to hear what all these wonderful scientists are working on. I was telling them that I think if I hadn't been an artist, I would have been a biologist. I was so fascinated, and it was a big surprise for me because I was always a kind of language person, but then I had an amazing teacher of biology, and then I had a kind of revelation that all knowledge was connecting, because when you study one cell and then just go on and on, building up to the human organism, and you see how everything is interdependent and how it builds, and that a lot of the same principles are the same. It's just so exciting to see the way that it works. The last piece that I did here at UCLA, as a matter of fact, was called On Behalf of Nature, and the last 10 years or so I've been really working on contemplating subjects or topics that you literally can't delineate into pieces, but the act of contemplating them is part of the process of creating a work. So it's not just making a piece or like a product, but it's really contemplating something that you'll never really find a form for it, but it's just, you show little glimpses of it, and the process also becomes part of it. So the first piece in that series was called Mercy, so how do you actually do a piece about mercy? Impermanence was one. And then this On Behalf of Nature, and I was very inspired by Gary Snyder's idea. Gary Snyder is a poet. He was a very early ecological thinker and also a Buddhist. And I was reading an amazing article that he wrote called Writers and the War Against Nature. And he was basically saying that the job of an artist is to articulate and to represent entities that don't have their own voices. And then he said, through music and dance. And I was like, ooh, this is very exciting. So that piece was kind of a contemplation of the energies that we are in danger of losing, rather than making it wasn't at all like a kind of direct political statement like, keep your plastic bags and don't throw them away, but it was more a kind of poetic meditation on really, in a sense, what we're in danger of losing. And after that, I became very interested in going deeper into that. And I started, usually if I make a large work for my whole ensemble, I work very quietly alone and usually do very small solo kind of pieces. So I was at the keyboard and I was working on some solo vocal pieces that I was laying tracks, so they were very complex, little tiny, tiny pieces. At the same time, I was reading Siddhartha Mukherjee's book called The Emperor of All Maladies, which is a history of cancer. And but what really struck me was his talking about the cell, and just as Meryl said, the mystery and the magnificence of the cell as a kind of prototype for cooperation and interdependence. And so I just started getting so interested in that, the energy of the cell, this kind of rotational activity, the behavior, and the interdependence of these elements. Then I was thinking how those really wove together as our bodies, millions of cells in our bodies, and that you could use that as a kind of prototype for a different kind of society, as a kind of prototype for cooperation rather than competition, for tenderness rather than aggression, so that was the beginning of this work. And as I was reading the book, I started thinking that my pieces, these little vocal pieces, had something in common with what I sensed as the activity, the motor of life, the motor of life, the activity of a cell. And also in musical terms, a cell is like a very small unit. It's an indivisible unit that you can build from. It's not like a phrase. You can actually have more than one cell in a phrase, but it's a rhythmic or melodic unit that's indivisible. So I started thinking, how that could actually be manifest as music and movement. And that's basically how the piece formed. So I guess we'll talk more about it in the panel, but I would like to show you a little tiny sequence from the piece. This was, yeah. So it's very complex, integrated, woven kind of vocal sound. Also, I was very interested in this rotation idea because I don't actually see 3 dimensions. So this is the first time that I felt that I was actually making a musical form that was sculptural. And so I really think of it as a sculptural, some of the pieces as sculptures, musical sculptures. And also, I think as artists now, all of us are asking what we can actually offer right now in this very dark world. So I also think of this piece as a kind of healing or a sort of antidote, and trying to make a piece about wonder in a world that's, you know, that we're really in danger of losing that sense of wonder and mystery. I don't know how much she left of this, and if it's too long, we'll have to stop it, but I'll see how long she gave you. Okay, all right.
Thank you. You're welcome. Thank you. All right, so next we have David Glanzman, who does research in neural processes and behavior.
Thank you very much, Rita. Well, that is a hard act to follow, particularly if you're a scientist. So I don't have any beautiful visuals like that. So what I work on, I'm a neurobiologist. I'm in the Department of Integrative Biology and Physiology and Neurobiology here at UCLA. And my interest is in the cell biology of learning and memory. And in particular, I'm fascinated by the question of how does the brain maintain memories over the course of years, decades, or in some cases a lifetime. And this is one of the most, I'm a little biased, but this is one of the most interesting biological problems I think there is. And I chose, following my mentor's steps, I chose to study it in an animal that many people would be surprised even learns anything, which is a snail. And the snail that I work on is shown here, a cartoon of the snail. And it's a marine snail, and it actually lives here in California. So if you are ever snorkeling in Malibu, you will see these snails, and they'll be in tide pools. And if you study them carefully, you'll see that they're eating seaweed. So you might not think that's very interesting, but if you spend long hours studying them very carefully, you'll see that they basically eat seaweed. But they also are capable of learning, and that's what I study. So what I study is a very primitive kind of learning called sensitization. And basically, this would be if you are exposed to some alarming stimulus, you will become hyper-reflexive to all stimuli after that. So this is a very primitive kind of learning. It's non-associative. But in these snails, they can remember for a very long time, depending on the experience. And so in these experiments, what we do in order to sensitize them is we give them sort of mild electrical shocks to their tail. And after that, they become hyper-reflexive. So if you were, after giving them the shocks, if you were to come up and touch them on their skin, they would contract very strongly. And that's how they show you that they've had this aversive memory. And for years, I've been studying this memory, and one of the common ideas in our field is that memory, when an animal learns something, there are changes in the connections that join the neurons, and they reorganize as a result of experience. And so for years, I have studied that, and I've been very interested in that. But recently, and this is kind of ironic, given that the theme of this is cells, this is recently I've become, my thinking has changed, and I've come to believe that the memory is not really in the synaptic connections. Really, it's in the cell, and it's in the nucleus of the cell. And somehow, the nucleus of the cell changes as a result of learning, and that persists. And it's independent, in a sense, of the changes in synaptic connections. So this is a radical idea in my field. Indeed, it's heretical, I would say. I think that's fair to say. But I wanted to try to establish this idea. And so we thought, well, what is it that is producing these changes in the nucleus? And many of you will be familiar from your freshman biology of DNA, of course, which encodes genes. And the way you get proteins is the DNA is transcribed into RNA, and then the RNA encodes for proteins, and that's pretty basic biology. But it turns out there are a whole new class of RNAs that don't encode proteins, and we don't really know what they do. And one of the things that these RNAs are involved in is changing the, producing nuclear changes, changing the likelihood that genes will be expressed. That's one of the things they do. So we thought, well, maybe what we could do is take a snail, train it, and then extract its RNA and inject it into a snail that has never experienced electrical shocks. And that snail would act as though it had been shocked. So that was the experiment that we did, and that's shown here. And to the astonishment of my lab, it succeeded. And so, so now we're trying to figure out just what is it that is changed in the snail? How does the RNA change what we believe are the changes in the snail? But the idea is, and I'm increasingly coming to this belief, is that it is perhaps the case that many of the neurons in our brains are unique, and they're unique because the experience has changed, produced intrinsic changes in the neurons. And so that's going to be something that's going to drive my research. And it's kind of ironic. I haven't even had that thought really until I was sitting here listening to Meredith. I'm thinking, well, that's what I'm moving towards, the idea that individual cells have their own unique identities, and that's due to their own unique experiences. So I think I'll let it drop there. Thank you. Thank you for your time.
All right, and then next we have Stephanie White, who does research with birds and bird songs.
Thank you. It's a privilege to be here with old and maybe new friends. Yes, so I'm also in the Department of Integrative Biology and Physiology, and I'm just really fascinated, not only as a neuroscientist in how the brain controls behavior, but I've just been really struck about how our behaviors, even our movements, our vocalizations can actually change our brains and change gene expression within the type of cells that I studied, the neurons in the brain. And to look at this, I study, and my team study birds, and not the scooter bird that you're seeing here, but actually songbirds. And what's been a privilege for me to discover about them is that when birds wake up and sing in the dawn chorus in the morning, they actually regulate the gene transcription in the neurons that are controlling that behavior. So it feeds back, the behavior feeds back and changes those genes and ultimately what proteins will be made. And so they're singing in the morning, and as they're doing so, they're listening to each other. And the type of neurons that they use to do this we call mirror neurons, because these neurons don't just help make their own behavior, but these neurons help them sense that behavior in another bird. So they're really helping the bird to communicate and to listen in. And so I find this pretty exciting in birds, and now there's even evidence in humans, especially gifted or professional performers who may perform on a concert, and then you can take blood samples from them and find that they're changing the levels of these same genes that we find in the bird in their brains. So I think that's really fascinating. And another sort of related aspect is that if you look at young children that have different abilities in language, including children that have language delays related to autism, and you look at their gene transcription in a group of genes that are, working together that are seen in the more autistic children, those genes have about 75% of our singing regulated genes from the birds in them. So the idea might be, when there's a social problem, when we're not functioning well socially and not able to interact with each other, that way we're not activating these cells and we're not making those genes to come back and sort of reward and get the brain in sync with others. So that's really a pleasure to be here tonight. Thanks.
All right, and next we have Dr. Elissa Hallem, who is doing amazing research in parasitic worms.
All right, thank you. It's a pleasure to be here. My name is Elissa Hallem, and I'm a professor in the Department of Microbiology, Immunology, and Molecular Genetics. And my research in my lab combines neurobiology and parasitology to try to understand the host-seeking behaviors of human parasitic worms. So we study these worms that are skin-penetrating human parasitic worms. They're found throughout the world in the soil, especially in warm climates, and they infect primarily when people walk barefoot through soil or are in contaminated water. They can also infect through water. And these infective worms enter the body by penetrating directly through the skin. And so this is what one of the worms, the infective larvae, look like. The species we study the most is called Strongyloides stercoralis. It's also known as the human threadworm. And like I said, these worms are in the soil, and they're actively sensing their hosts. And so that's the process that we study. And so if there's a human standing here, these worms are sensing cues that are given off by the host. And these cues include heat given off by our bodies, also odors. So we emit hundreds of different odors from our skin and sweat, and these are sensed by the worms. And the worms are also using these odors to distinguish a human host from, say, a cow or a rat, which they are not able to infect. And so they're using odors to try to discriminate a host, like a human, from a non-host. They also sense vibration as we walk by, taste cues, so many different cues. And so my lab is interested in understanding how these little worms in the soil integrate all of these cues that are coming off of a human and how they use these cues to distinguish a human from a non-human. And in particular, we're studying the neurons in the worm brain that detect these human cues, how those signals are integrated in neural circuits that tell the worm, okay, now it's time to actually penetrate into the foot and infect. And we're also interested in the molecular mechanisms that mediate these processes.
All right, and last but certainly not least, we have Dr. Christopher Colwell, who is looking at circadian rhythms.
Yeah, well, thank you. The wrong way. Good. Okay, so just a moment to define what a circadian rhythm is. These are, I've lost myself. These are endogenous rhythms with a cycle length of close to but not equal to 24 hours. So they govern all parts of our body. So not just we've heard about nervous system, but also everything from your heart, liver, lungs, the cells in your skin. Everything are governed by these endogenous rhythms. There's been a number of what I consider key discoveries in this area over the last few years, one of which is sort of illustrated in this cartoon, which is understanding the molecular genetic underpinnings of how these oscillations occur. So in this case, I have, this prototypical cell, but it could be any cell in your body, okay, with the nucleus we've heard about before, and then the cytoplasm. And at the beginning of the day, so think about this as dawn, there's the activation of a set of genes, shown here is the period gene and cryptochrome gene, and the mRNA is produced. Yeah, it gets translated into proteins, which are out in the cytoplasm. They accumulate over time, over hours, okay, and then near dusk, they're translocated back into the nucleus where they turn off their own transcription. So that's a negative feedback loop that takes 24 hours. And that's what governs these rhythms in all of us. So some of you are morning people, you have a shorter molecular clock. Others of you are night people, you'll have a longer molecular clock. And I could take a skin sample from you, grow these cells in culture, and you could see the molecular genetic underpinnings of this part of your behavior. But it doesn't just regulate when you go to bed or when you wake up, but actually, again, everything in your body, heart, liver, lungs, they all have to be coordinated in time. And this system is occurring in all the cells in your body to do that synchronization. Yeah. So just a couple other points about this system. One of the other key things, besides understanding the molecular genetic underpinnings, which, by the way, the work leading to this was done originally in Drosophila and was awarded the Nobel Prize last year for discovery in medicine. Okay, but the other thing which has been, I think, a major discovery from this area of research is that we have a separate sensory system in our retinas that detect the light into this clock. Because what good is a timing system if it can't be synchronized to local time, right? So just in the same way you have to reset your wristwatch or whatever. Well, I guess we don't have wristwatch anymore. If we do have wristwatch and you reset it regularly, okay, this is the way our body has to be, the clock in your body has to be reset every day. And light is the cue that does that, but it's not from the rods and cones that are part of the visual system. There's a separate light detection system, which is actually really cool on a molecular level. It's very similar to what's found in a fly. So even it's a very ancient system. And it's very relevant to our world right now because it's a little bit hard to see here, but it's the blue-green wavelength light that this is sensitive to. And most young people that mess up their sleep-wake cycle and have problems sleeping, it's usually because of inappropriate light exposure. Because there's, right now, we're all sitting in the dark for the circadian timing system. It is only sensitive to blue-green wavelength lights, and there's very little in this room right now. So if your brain says, okay, this is dark, your visual system tells otherwise, but for this clock, it's dark right now. And yet, if you then went home and played a video game or maybe you read with an electronic reader, that emits blue light right into this system. and that will cause your brain and the rest of your body to start following light, and your sleep-wake cycle will start getting inverted. So this has been a major discovery in the last decade or so. And finally, the final point I would like to make is how this system works. Okay, so I already mentioned the molecular clock is found in all the cells in our body. Okay, but the light detection system is in our retina, okay, not in the rosin cones, but in a separate cell population. There's a central clock in our brain known as the suprachiasmatic nucleus, and then it sends out signals, both hormonal and neural signals, that then communicate the molecular clocks found in all your major organ systems. Okay, so this is the way the system works. It's a distributed network of oscillations. So each of these tissues have their own oscillation, and the set of genes that are controlled in your heart are different than the ones in your liver or in your skin, for that matter, or whatever your favorite bit is. Okay, and so this is a system which gives a lot of flexibility. It's also sensitive then to perturbations that we do to it by working in appropriate shifts, by jet travel. These are all, if you've experienced this, you know exactly what it means to have your circadian system disrupted. And increasingly what we're finding with aging as well as diseases of the nervous system is that this system is disrupted. And so we're focused then on trying to figure out new treatments to help with that. And that's it. Thanks.
All right, I'm so excited at the very broad diversity of this panel. So I'm going to invite all of our panelists to come and have a seat, and we're just going to have some informal chats. Here we go. All right, great. So even though everyone's research or artistic practice is a little bit different, I think there is one theme that does communicate, or rather one theme that does go together with what everyone has been doing, which is communication. That everyone's work does, on some level, in some way, involve communication. So I guess just to start us off with maybe sort of broader question, how do you each define communication in the context of your specific field of work? And this question is for any of you.
Well,
Slash
okay,
all of you.
yeah, okay, yeah. So, in my system that we study, we're talking about a system of oscillators, right? So even though each cell has its own oscillation, its own molecular clock, they all have to work together for the system to be in place. So there's communication, but the difference for the sort of system that we studied is that it's very slow. So most of the timing in the nervous system, they talk about in millisecond communication. But the things that we study are over hours. And so we study processes of communication that take much longer, that involve changes in gene expression inside the cells. So that's how we look at it.
Yeah, and I would say in the songbird, it's pretty analogous to the human, how we think of communication, right? That the song is used to, in some cases, court or alert the other organism to the presence. So there's that, and there's also another aspect of it, which is a shared attention, a shared joint attention and an appreciation for, what's going on, and an awareness. So I'm really about sort of more social, an organism-to-organism type of communication. But of course, within the brain, the neurons that are participating in that need to communicate with one another, and so they have to both have the sensory input of hearing the signal in the environment, and as you go up the pathway from just the ear, where the neurons are going to fire for any sound between 2 and 2 kilohertz in humans. As you progress up, you get to these neurons that only respond to the bird song. They don't care about any other any other sound. So that's very interesting to us how that gets shaped. And then those same neurons in turn communicate with the behavioral output to sing that song. So both the interorganismal and also within the circuit, those communication signals.
I feel so related to what you're saying, actually both of you, because as a performers, that's exactly what we're doing, because we're, as I said before, the music is so complex and interdependent that in the forms themselves, there has to be this kind of sense of interdependence to even perform these pieces. So there has to be that same sensory alertness and awareness, and so we're communicating between ourselves, but at the same time, that vulnerability and that immediacy, we're trying to communicate that to the audience as well. And I think with my work, which is very abstract in a certain level, I think that we're communicating through different code systems, emotional, kinetic, sensory, intellectual on a certain level, but in a way I'm asking for a little bit of the discursive mind to take a rest during these pieces. But I think that, so it's very instinctive on a certain level, but what I like to do with an audience is never to feel like we're manipulating them in any way, but to make it that each person feels that they have their whole being intact as they experience this. So it's really offering a kind of direct experience that people we're not leading them to, it's more like poetry in a certain level, that each person can have a unique experience of something. And so that also hooks back to their own lives and memories, and so it makes it very personal, and at the same time, I'm trying for something that's also very universal.
Okay, so in my research, we're looking at, I guess, sort of accidental communication, maybe, between a human and a parasite. So, so these parasites are found everywhere, and they infect over 1 billion people worldwide and cause extensive disease. And as I mentioned, they're actively sensing their human hosts. And so I'm looking, humans give off, like I said, all of these different sensory cues, and we're looking at how that's communicated to the parasite and how that drives the parasite to find and infect a host.
So when I think of communication with respect to my work, so I think of several types of communication. So first of all of us are unique, and in part we're unique because of our experiences. So communication is a way that the environment sculpts us to a certain extent. So the environment gives us rewards or punishments depending on what we do. So that's a form of communication. And then, of course, within the nervous system, there's a rapid form of communication, the way neurons communicate with each other via synapses, and that's a very rapid and pervasive form of communication. But it's interesting. So if you go back 100 years or so, there were two great neuroscientists, one by the name of Ramoni Cajal and the other by the name of Camillo Golgi, and they had very different ideas about the nature of the nervous system, and they hated each other, basically. Ironically, they were both awarded the Nobel Prize in the same year, and so they had to be on the stage with each other. So that must have been interesting. I wish I could go back in time and see that. But Cajal had the idea that that the nervous system was made up of individual neurons, and they were separated by gaps, which we now call synapses, whereas Golgi had the idea that the nervous system was a big syncytium, a sort of network, and there were things flowing all over, and that the idea that it was individual cells was, I think he thought, was more or less an illusion. Well, fast forward, everybody today believes Cajal, but there's increasing evidence that neurons may be communicating by exchanging molecules, some of which are RNA, and this may be a very slow form of communication, very ancient. It may antedate the nervous system, in fact. And so this is, we don't really know if indeed this is going on. We don't have a very good grasp of what the neurons are communicating to each other. So that's something that I'm very interested in.
We will have time at the end definitely for audience Q&A. So hold your question. Yeah, write it down so you'll remember it, because we definitely want to get to that too. But I have a question for you, Meredith. So we've been talking about communication, and there's just been, again, sort of a theme of kind of hierarchical structures, both in cells and in communication. So you mentioned your use of musical cells in your piece. Do those cells in your piece, do you think, come together to form like hierarchical levels in the same way that cells in our bodies come together or work together to make a sort of larger organism? Was that something that you were thinking about in your work? Or what themes specifically with cells did you use when you were constructing your musical cells?
I mean, it's not a one-to-one relationship. You know, it really can't be. You know, otherwise you're just making a kind of arbitrary system that you're fitting music into, which some people do. I mean, that is a methodology, but like that's not my methodology because I work much more organically. So I don't think I would have ever thought of it as any sense of hierarchy. It's actually, you know, I wasn't even thinking of it that way particularly. It's much more, I was thinking in a sense the kind of, the energy and the three-dimensionality and the processes that are similar, you know, between cell behavior and musical behavior. So, you know, but I was thinking, you know, I could never, my inspiration could never be that direct from a system like that. You know, music is music, so to speak. So I never thought of it in terms of hierarchy at all. I thought of it much more as very intense weaving and beading and layering and replication as like that replication is a cell activity, but that's also something that's also intrinsic to music. You know, mirroring, it's interesting that you were talking about mirroring because you'll see the film that we have is, it's kind of, there's a mirror kind of film images on the floor and on the back wall, and so that this mirroring kind of idea was definitely part of what we were working with. So it's these principles, and also I think I was trying to get something that going from the microcosmic cell to the macrocosm of space and the universe, thinking that probably I was just going blah, blah, blah because I'm not a scientist, but that cells are in the universe is what, you know, so I was going for the smallest unit and then how that actually, those same processes go out into space. Then there's a whole nother layer that, you know, I guess I'll mention, even though it probably has nothing to do with this discussion, but is that there was a certain, I have a vocal ensemble of both men and women, and they're wonderful, wonderful musicians and singers and dancers, I mean, unbelievable. But there was a kind of intuition that I had about, it was 2014, that this kind of patriarchal impulse was coming up very strongly. It was just an intuitive sense. And so I decided that I wanted to do, and also I think it had to do with my first pieces that I was laying tracks of my own voice, so they were treble voices, you know, rather than all the way from top to bottom, so close to the bird songs. But so I just decided that it seemed really important this time to work just with a cast of all women. So it's a 5-woman piece, each one very unique and strong and very much themselves. And then all the male members of my ensemble were like, oh, we're not going to be in this piece. I said, you know, I just explained. They said, oh, we understand, Meredith. We'll wait till the next one. But that's also a very important thing. And also, after I started working on the piece, I also read another Siddhartha Mukherjee book, which is called The Gene, which I plowed through. That was fascinating. And so if you see this in the lobby, try to find it if you come. There's an installation that I made of the 5 of our faces. So and it's 5 monitors, just on little tripods. I wanted it to be very simple, not arty or anything. And you see the 5 faces, and each face is very unique. And there's some of the music is almost like passing, like we call it hocketing. So one person has one note, and each person would have a cell. You could call that a cell. And by putting those notes together, you get a melody. So one person is only in charge of one note, but it makes a melody as you pass it on. So you see the faces, and we're singing one of these pieces, but sometimes you'll see a close-up of our mouths. Ha, 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 ha. And you just see our mouths, or you'll see like our eyes. And then because we come from so different backgrounds, I felt that what I didn't get into the piece was this idea of DNA, like our DNA component of our cells. And I couldn't get it into the piece. The piece is, you're far away from us as performers. So I wanted to introduce that aspect, that you could see these 5 faces, and then there's a section where you see things like MRIs, our own personal MRIs or X-rays, so you start seeing in the body. And then there's another section where you see photographs of our families, starting from our parents, going back to as far as we could go. So I got even my great-grandparents, I didn't even know they existed. So that was like a process of the piece, each person finding these photographs, going back in generations to, in a way, sort of acknowledge our ancestry. And that's why I was asking David what he was talking about. Is that epigenetics? Because that's also such a fascinating concept. I've also read Bruce Lipton, who probably you think is a dodo on some levels, but what he was talking about, how the environment is also so important in relation to our cells, that his experiments were actually eliminating the nucleus completely, and the cell still was like taking in information. So I'd love to actually hear what that in other words, what he was saying is we are not imprisoned by our DNA. That's actually what I got out of what he was saying, is that now, there's so many, that we think if this DNA exists in us, this is our destiny. But that's not necessarily true, that it's really, really changed and modified by our environment.
No, that is
So,
true, yeah.
I'm sorry, blah, blah, blah.
No, it's great. So this next question is for David. So you mentioned the types of stimuli that you give to snails.
Yep.
I know that you also study zebrafish, which is another common animal model.
Yes.
What type of stimuli would you give to a zebrafish? And with the changes that it makes, are those changes ever generational changes? Like, do you ever see them then in subsequent lines of the animal?
Yeah, so we have not studied that. I'm sorry to say. So we only look at within the lifetime of the animal. So we've not looked to see whether you can get epigenetic changes that are passed on to the next generation. I know there are people working on that, and they're trying to provide evidence that can happen. Most of those people are working on mice, but I don't really look at that.
So what type of stimuli then would you give to, say, a zebrafish, since that's another one that you look at?
We're interested in very, very simple kinds of behavior and very simple kinds of learning. That has to do with my bias as a scientist. I tend to be what's called a reductionist. So I want to take a complex phenomenon and try to reduce it to its essential elements. And so with the zebrafish, I don't know if any of you have ever tried to grab a fish in the water. I'm a fly fisherman, and first I try to hook them, and then when I get close, I try to grab them. And if you ever try to do that, what you'll see is, what you think is happening is the fish is so fast that it just swims away from you. But in fact, that's not what a fish does. What a fish does is, as soon as it sees your looming hand or feels, if you manage to touch its skin, it has these giant neurons in its nervous system that cause it to contract very powerfully. And that scoots the fish away from you. And you don't even, that reflex is so fast you can't resolve it with your eye. And then it swims away. That's why you think it's swimming. That's the only part you see. So what we look at is we give these loud sounds, and we look at the fish give that start. And we can't see it, but we record it with a video camera. And so we're interested in how the escape of the fish changes with learning. So that's what we're interested in.
Do they have ears? Do fish have ears?
They have lateral lines, and they do have ears too. Okay. Yeah, they can sense. They have sort of vestibular-like organs along their body, and they can detect. They sense with that too. But they also have ears.
Very cool. So Elissa, this question is for you. So in your case, I guess, in a lot of cases we're talking about maybe improving communication. In your case, we're talking about stopping communication or trying to subvert communication. So I guess with parasitic worms that infect you or attack you if you're barefoot, probably the most common way to stop it is to just have a physical barrier. But what other ways could you stop this type of communication from happening?
Right, so one that we're thinking about is the potential use of like a trap or repellent. So that's widespread obviously for mosquitoes, right? Mosquitoes are similar in some ways. They can smell you and move toward you and sense heat coming off your body. And mosquito traps or repellents like DEET are very widespread, but this is not something that has been looked at all in the context of parasitic worms. And so you can imagine that if you could prevent them from sensing you, then you could prevent infection in the first place. So the issue people have is you can wear shoes, but in endemic areas, the worms are everywhere in the soil, right? And these are also very low resource settings where most people don't have shoes. And if you get infected, you can take a drug that will clear the infection, but a couple months later, you'll get infected again. So the drugs that we have don't prevent reinfection, and so that's the problem. People are just getting infected constantly. And so what we found is that you mentioned a hierarchy of cells, I guess, before. So we found that worms use a hierarchy of sensory cues. And so, for example, from a distance, they'll sense heat, and they'll move very quickly up a temperature gradient until they get to human body temperature. And then at close range, they sense odors. And that helps them actually say, okay, this is a human. So you can imagine like a heat and odor-based trap, for example, or a chemical that will prevent the worm from identifying you. So that's sort of the long-term goal, but this is at the very early stages of trying to understand what they're sensing.
Interesting. Stephanie, question for you. So you discussed mirror neurons when you were giving your introduction. Does that at all with the songbirds connect to any sort of sense of empathy that you could detect in these birds?
That we have not looked at that, not in the type of empathy I think that you're getting at, but we do know, your typical lab rat does exhibit empathy. And whether they're using mirror neurons for that or not, I'm not really sure. They could be. So this idea of mirror neurons, we have a local expert here, Marco Iacoboni, who's studying them in humans. But they're sort of the monkey see, monkey do neurons where, as I said before, they were thought to just be motor, so people were studying them just because if they fire, then you're going to get this interesting behavior. And then when the organism observes that behavior in another organism, they also fire. So they could be the basis of empathy. And so I know how you feel when you go yow, because I know how I feel when I go yow, right? And so they're mirroring in that regard. So we know our birds have those in the vocal domain, and they have them also, they probably are good for alarm calls like, yikes, you're making that sound, there must be a danger here, or something like that. Mm-hmm. But I don't think anybody's been looking at them in the cases of the empathic rat yet, but it would be interesting, yeah.
Very cool. And then my last question, and then we'll open it up to everyone, is for you, Christopher. So you were talking about cycles, obviously, with your work. So how challenging is it for the entire system to adjust to a brand new cycle? Like if you were suddenly thrust on a different planet where the light cycle was entirely different, how does that process from cell all the way to entire body actually change?
Right. So luckily it's an adaptable system, but there's a range in which we can synchronize to. So it's very good for around 24 hours. You travel to Europe, you travel to Asia, the system adjusts to this. There are some cycles, though, that are very hard. I don't know if any of you followed like the Martian landing, and they had interviews with the scientists as it was going on. And so the scientists were trying to keep on the Martian schedule of 22 hours. And it's beyond the capacity of our system to synchronize to that well. And so as you sort of watch the scientists going through reporting on this, I mean, they stopped it after about a month because they were all, crazy. But you could sort of watch them going down this path. It was a lot of fun for me. I could have told them it was a really bad idea, But in general, so it's just the experience of jet lag. It's the experience of So your brain adjusts, let's just say we're going to go to Beijing. Okay, your brain will adjust within 2 days, more or less. But other parts of your body, your heart, your liver, take a lot longer. So the liver may take 2 weeks to adjust to that same time. So during that interval of 2 weeks, you have the experience, and we've all, probably most of us have had this experience, of the brain being on one time, the liver being somewhere else, the heart somewhere else. And you know, then you have this set of feeling of a little bit of mental confusion, a little fog. Okay, that's on the mental level, but you also see cardiovascular symptoms, and of course the GI disturbances that you know probably most of us have experienced at one time or another.
So it's not a simple case of like top-down adjustment. It's maybe more of like an individual sort of bottom-up pieces that eventually maybe you'll adjust to.
Yeah, the brain is the unique part that it gets the light information directly. Now in flies or some other organisms it's different, because then any of the cells in the body can actually respond. But for humans and mammals, the light has to come in to this path that goes into the central clock in our brain. And that's relatively quick to adjust to the new time zone. But then that information about the new time has to be communicated to all the other cells in your body. That can take weeks, depending on how far.
Very cool. All right, so let's maybe take the last 5 or 10 minutes or so of our time to open it up to some Q&A. And this also, I will say, includes the panelists. So if you guys have questions that you would like to ask each other too, that would also be very interesting. But audience or panelists, if you have questions. I know that we had one question.
So about the snails.
Oh, could you, sorry, one sec. Could you use the mic so that, yeah,
So
thank you.
about the snails.
Yes.
Can you undo it and give them a soothing one that's already been stimulated to overreact? So can
That's a
you?
great question. So we are trying to do that now. So there's another kind of learning that you can, that's called habituation. So that's the type of learning that you start ignoring stimuli that are constant and unchanging. So for example, you're wearing a jacket right now. You're not feeling that jacket until I said, oh, you're wearing a jacket now. Now you can feel it. But that's because your body, your brain is habituated to that sensation. So the idea would be what we're doing now is we're subjecting snails to a long series of boring taps on their skin, and then we want to try to induce habituation, and then we're going to see if we can transfer habituation memory, and then we want to see if we can take an animal that has been sensitized and inject RNA from a habituated snail into that snail and see if the sensitization goes away. So it's a great question, and we're
Well, I'm kind of interested in because of treatment for trauma. So
Yeah.
I'm an addiction
Yeah.
mental health counselor,
Yes.
and I'm an artist.
Yes.
And so, you know, the
Yep.
whole theory, trauma stored in your body. But what you were talking about made me think that some approaches where you are flooding people with PTSD to have them to relive the experience may not be a great idea on a cellular level. I mean, you're talking about
Well,
that these things are happening.
There are very, there are many experimental approaches to PTSD that people are trying to use. I don't know if you're familiar with reconsolidation blockade, so that's another one that people are trying. And I used to believe that you could erase memories, but now I don't believe that. I think that certain forms of memory are quite intractable because they're actually involve epigenetic changes or maybe changes in the structure of DNA. So to remove a traumatic memory is going to be unfortunately, I think it's going to be very, very difficult. And in principle, it would require something like going in and changing the epigenetic changes, or perhaps, trying to reverse changes in DNA, something like that. So I'm actually kind of pessimistic. I'm much more. If you would ask me this question 10 years ago, I would have said, no problem. We're able to erase memories in snails. We can do it, and just upregulate to people, and blah blah blah. And I think it's actually going to be tremendously difficult to do.
We have a.
So how long do you think until the research on the snails and the songbirds would be used on humans? And why would it be used?
So some research on songbirds is already being used on humans. So in trying to figure out what the different parts of the songbird brain control of the song, a scientist actually put a little cooling probe on a part of the bird brain and showed that it slowed down the song. And then that same scientist went to neurosurgery that was being performed on a human who had epilepsy, and the surgery is trying to remove the part of the brain that causes the epilepsy. So to test to make sure that they're not taking out the part that helps them speak, they cooled the different part of the human brain. And just like in the bird brain, when they cooled one of these parts, the person was being asked to say Monday, Tuesday, Wednesday, Thursday, and they started going Monday, Tuesday. So we really think they're very similar, even though there's no way that there was a common ancestor that gave rise to bird song and human speech. But we actually think that we can learn a lot about what speech is doing through the birds.
For the, I think she asked also for the snails. Is there going to be an easy way to translate the snail research then into human research, or is that going to be a big leap?
It's a big leap. Yeah. I mean, it's really interesting. The response to our paper among scientists has been sort of skeptical, but the response among the population has been overwhelming. And it's because everybody's fascinated with this idea of memory transfer, and it's in all the movies. And sometimes I get calls from people in Hollywood who want to talk about scripts and things. But it's important to understand that this is a very simple form of memory in a very simple organism. So for me, it's not so much an approach to doing this in people. It's really what the research tells us about the nature of the biology of memory. And I'm under no illusion that this is going to be something that'll be immediate. Well, even in my grandchildren's lifetime, if grandchildren exist in 100 years, which I'm not sure about, whether or not there's going to be anything like memory transfer possible in people.
I'm responding just from my own experience. My music is written for the human voice, but it doesn't have language in it. It's all nonverbal language, although I always feel like it's right at the edge of language, because I've always believed that the voice itself is a language in itself. So when you put English on top of it, it's actually two languages simultaneously. And so something about the bird communication, I feel very close to, because we can go around the world and people respond really directly to this music because they don't have to go through the filter of language to get to the kind of codification and the communication that we are actually in the act of doing, as well as communicating to an audience. So I feel very interested in the in basic utterance, and the beginnings of, and there's some very interesting experiments that they've done about music in that. For example, there was one research paper that I read that they thought that ancient people actually all had perfect pitch. And when verbal language came in, then some people lost it in their brains. And actually, we have one person in my group that does have perfect pitch. And so in a way, she has a more ancient brain than we do. But, and then there's the other strange thing, too, that in Asian cultures, something like Chinese culture, which is like Mandarin, they did some experiments where they had somebody say something and then a week later say the same thing, because it's a pitched language, and it was absolutely like they could measure it by, absolutely to the exact decibels, and it was, the exact sound. So then there are more people in the Asian world that speak the sung languages, and they also have the more absolute pitch. So I think it's very related to the birds somehow.
Yeah, absolutely.
Yeah.
And in the species that we study, which is not a zebrafish, but a zebra finch, the adult male, unlike, in mockingbirds or parrots that continue to mimic throughout their lives, when the male gets sexually mature, he's got his courtship song and that's what he's going to use. But we could put little headphones on him and play back his song to himself, and using a harmonizer to make it sound like it's going up a little bit, and then he'll start singing the song deeper so that he can bring it back, and he knows exactly where that pitch should be. And you can punish it by little tiny amounts. You could just give him a little you give him a little white noise if he sings it just a little bit outside of the frequency. And they will adjust to avoid that. And as soon as you get rid of that white noise, they go right back where they want it to be.
I think that all your research is interesting.
It sort of also reminds me of going to Europe where A is 448, and in America, the frequency is A440. So I can actually go. I remember going to Europe the first time trying to sing with an A448 piano, and I could actually feel it in my, even though it's so tiny, I could feel the difference in my vocal cords, how much I had to have a little bit more energy and tension to get that to be in, it was just that little tiny thing made a big difference. So, and probably the male bird it's a feeling thing as well, it's also a muscular sensation, to make it that precise.
Great. I think we have time for one more question, and then we'll have a sort of maybe informal chat outside. So please.
I was interested in this previous comment that communication sort of goes both ways, right? So behavior affects the brain, affects behavior. I talk to you, you talk back to me, I need to be attentive. And I was wondering, for the performing artist, you're performing a piece, and I get it, but you get nothing back from me. So, or do you?
I always think performing is like an infinity sign. We're sending out, I think of it more in terms of energy. So we're sending out energy, and then it's coming to you, and you definitely are sending us back energy. It might not be verbal energy, or we're not having a direct conversation, but we're having a conversation. It's, we always, and it's not that we want to be loved or anything. It has nothing to do with that, that instant gratification. It's much more that sense that we all are in this same energy field, and you really can feel the difference between when that happens and when it doesn't. So that's what we're trying for, that we are getting energy back from you, and then that's feeding our energy, and it just keeps going around like this. So that's what a wonderful performance is. Plus, I think that the thing that's beautiful about live performance that you cannot get on the screen is, our sense of vulnerability, and that we're on a kind of tightrope, and we can fall off at any time, and that. So you actually can feel that experience, that immediacy, and I think it's something that doesn't happen in too many other places in the world that we're living in now, and you could never really get that on I love movies, I'm not saying I don't love them, but it's very different. So it's more a sensory and kind of energetic and kind of sensation.
And again, a social aspect of the communication that you wouldn't get from our gizmos.
Right, exactly. Mm-hmm.
I would also just add one thing myself personally, too, that as a performer, it really also depends on what you're performing and what the normal cultural expectation is around that performance, too, because in some performances, you're performing and the audience is sitting quietly or intently and watching you. In other types of performances, the audience is shouting and cheering and totally singing along with you, and then it really does become a very sort of two-way exchange of energy. So I think that also depends as well. But thank you all so much. Well, first, thank you, audience, for being here today to have this conversation with us. And thank you all panelists so much. This was a really very wildly diverse and really incredible conversation that we got to have. And so hopefully now we can all maybe get up together and enjoy some refreshments outside and maybe continue some conversations. And I also want to thank Meryl, who helped put all of this together and again was sort of the instrument in bringing us together with Meredith and these wonderful scientists. So thank you so, so, so much. And thank you, everyone.